Today we will be discussing about acquired immunity. Unlike innate immunity, acquired immunity is not present right from the time a child is born. This kind of defense system, it is acquired by the body when the body is exposed to different kinds of pathogens. So what are the chief components or the chief cells involved in acquired immunity? So there are three chief cells which are involved in acquired immunity. The first is APC, the second is B cells, and the third is T cells. APC stands for antigen presenting cells, and these include monocytes, dendritic cells, and B lymphocytes. So B lymphocytes or B cells also come under antigen presenting cells. Okay. Now all these three cells, they are formed in the bone marrow. And bone marrow is the primary lymphatic organ. So first talk about APC. APC, that is antigen presenting cells, they are formed and matured in the bone marrow, and they also proliferate in the bone marrow. Proliferation means they make multiple copies of it. They divide and they make so many copies of it. Okay, so the formation, maturation and proliferation of APC take place in the bone marrow, that is the primary lymphatic organ. From there, it is translocated to different parts of the body. As far as APC is concerned, they are translocated to those parts of the body which are the entry points for pathogens. So their translocation occurs in the skin, then mucous membrane, then urinary tract, respiratory tract, as well as reproductive tract. So these are the entry points for microorganisms or pathogens. Now let's talk about B cells. B cells are also called as B lymphocytes. Their formation, maturation, proliferation and differentiation take place in the bone marrow. So the formation of B cells, it takes place in the bone marrow. Then these B cells mature. After maturation, they will divide. They will make different copies of it. That is called proliferation. After making different copies of it, they differentiate. Differentiation means each B cell differ from other B cells. So all these take place in the bone marrow. After that, it will be translocated to the secondary lymphatic organs, that is spleen, lymph nodes, tonsils, and Peyer's patches. These are the secondary lymphatic organs. Bone marrow and thymus, they are considered as primary lymphatic organs. Now why B cells are called B lymphocytes? What this B stands for? It's not that since it is formed and matured in the bone marrow, that's why it is called B cells. No. B cells were first discovered in a specialized organ, and that organ is present in birds, and that specialized organ is called bursa of Fabricius. Bursa of Fabricius. It is a specialized organ that is present in birds. That's where B cells were first discovered by the scientists. And that B stands for bursa, just to signify the importance of this organ. Okay. Now let's talk about T cells. T cells are also called as T-lymphocytes. Formation of T cells take place in the bone marrow, but its maturation, proliferation and differentiation, it takes place in thymus. So from the bone marrow it will go to thymus, and here T cells will mature, proliferate and differentiate. After thymus it will go to the secondary lymphatic organ, that is spleen, lymph nodes, tonsils and Peyer's patches. Okay, so these are the three main types of cells which are involved in acquired immunity. Now there are two types of acquired immunity. First is humoral immunity and the second is cell-mediated immunity. Humoral immunity, it is associated with antibodies, and these antibodies, they are present in the body fluids. As far as cell-mediated immunity are concerned, they are present in the lymphatic organs, and they are associated with this kind of immunity is associated with T cells only. So in the next section we will be discussing about humoral immunity, and after that cell-mediated immunity. So humoral immunity is also called as antibody mediated immune system, and as I said, antibodies are present in the body fluids. So how this immune system works, that we will see in this section. So the first thing that happens in the humoral immunity, it is the encounter of the pathogen with the antigen presenting cells. That is, encounter of pathogen with APC, antigen presenting cells. Here I am taking pathogen as bacteria. So I am drawing a bacteria. This is a bacteria. Now I am drawing a macrophage. So once this macrophage encounters a bacteria, what it will do? It will phagocytize this bacteria through endocytosis. So first thing that happens is endocytosis, and then phagosome will form. Inside the phagosome, bacteria is there, and in this macrophage, the cytosol consists of lysosomes. So these are the lysosomes which are present in the cytosol. These lysosomes will attach to this phagosome and form phagolysosome. That we have already seen in the innate immune system, how phagocytosis takes place. Now after this all the content in the bacteria, it is digested by the hydrolytic enzyme. Now in phagocytosis, if you remember, once the pathogen is digested, it is removed from the cell through exocytosis. But here in case of APC, antigen presenting cell, it does not happen. Exocytosis does not happen. What happens here? Once the content is digested, the antigens float in the cytosol, and in APC there is a special molecule present that is called MHC2 molecule or MHC class 2 molecule. MHC class 2 molecules are present only in antigen presenting cells, that you have to remember. Okay, now MHC2 molecules, these are MHC2 molecules which are present in the cytosol. The function of these MHC2 molecules is that each MHC2 molecule will take each antigen on the plasma membrane of the antigen presenting cell. So what this will do, it will take each antigen on the plasma membrane of the macrophage. Now we can call this macrophage as antigen presenting cells because this cell is presenting antigen on its surface. This is the first thing that happens during in humoral immunity. And it is important to note that APC, antigen presenting cells, they are located on those sites in the body which are the entry points for pathogens like skin, mucous membrane, urinary tract, digestive tract, respiratory tract or reproductive tract. So whenever a pathogen enters into the body, it first encounters with the APC. Okay, so this is the first encounter of pathogen with APC. Now what will happen? This APC is present at the site of infection. Now this APC will go to the lymphatic organ, nearby lymphatic organ, means the lymphatic organ which is present near to the site of infection. So this APC will reach to the lymphatic organ, say lymph node, and we know that in the secondary lymphatic organs we have B cells and T cells. Okay, now what will happen? This APC will encounter first, it will encounter with T cell, and that also T helper cell. Now this T helper cell, this is a T helper cell which we denote as TH. Now this T helper cell has TCR, T cell receptor. What this TCR will do? This TCR will combine with this antigen. Okay, so they will form a complex here. This TCR will form a complex with the antigen, and there is another molecule which we call CD4 molecule that combines with MHC class 2. These are MHC class 2. So MHC class 2 will combine with CD4 and TCR will combine with the antigen. Once this combination is formed, this APC will secrete IL-1, interleukin 1. Once this interleukin 1 reaches the T helper cell, the T helper cell secretes IL-2. The T helper cell secretes IL-2. Once the IL-1 sends message to the nucleus, the T helper cell also secretes IL-2. And because of the secretion of this IL-2, the TH cells will start division. The TH cell will proliferate. So many T helper cells are formed. So many T helpers are formed, each having TCR, and these TCR, each T helper cell has a TCR, and that TCR will recognize only this antigen. Okay. Now, up to this point, everything is clear. Now, we have another B cell. As I said, in the lymph node, if APC approaches T cells, so there are also B cells present. But if you remember, I said that B lymphocytes or B cells are also considered as antigen presenting cells. So, like macrophage, B cells will also behave like antigen presenting cells. So whatever happened here will also happen with the B cells, because B cells are also considered as antigen presenting cells. Means B cells will also contain MSC class 2 molecule. B cell will also encounter with the antigen. It will also phagocytize the antigen. It will also present the antigen on its surface using MSC2 molecule. So let's see, this is B cell. I am not showing how phagocytosis took place, but this B cell will also have MSC2 molecule, and it will also represent the same antigen that this APC encountered, the same antigen that T helper cell encountered. So B cell will encounter with the same antigen, and because of the presence of MSC2 molecule, it will also present this antigen on its surface. Now this B cell will get attached with the T helper cell. So let's see what happens when T helper cell attaches. This is T helper cell, this is B cell. Okay, when this T helper cell attaches with the MSC class 2 molecule and the antigen, it will form a complex here also, and this T helper cell, it will secrete IL-4, IL-5 and IL-6, interleukin 4, interleukin 5 and interleukin 6. Once this is secreted by the T helper cell, the B cell will proliferate. It will start division. Okay, Now B cells will form so many copies of B cells are formed. Out of these cells, B cells, some will differentiate into plasma cells and some will differentiate into memory cells. So two types of differentiation takes place: plasma cells and memory cells. Now this plasma cell will form antibodies at the rate of 2000 per second, and these memory cells, they will just circulate in the body fluid. Okay, now what these antibodies will do? These are the antibodies, they are Y separate. We will see the class of antibodies in our later lecture, but here we have to focus on what these antibodies do. So once these antibodies are formed in the lymphatic organ by the plasma cells, these antibodies will circulate in the body fluid. For example, these antibodies will circulate in the blood. Okay. Now, if antigens are present in the blood, or in the form of, or the pathogens are present in the blood, and as we have said, we have taken an example of bacteria. Suppose bacteria, they are present, they are circulating in the blood. So what these antibodies will do? They will attach to the bacteria, okay, and they will immobilize this bacteria. Once this bacteria is immobilized, the NK cells or the macrophages, they will eat this pathogen easily because it is immobilized. It is not circulating with the flow of the blood because it is bonded with the antibodies from all the sides. Besides, we also know that we have studied a complement system in the innate immunity, and as I said, complement system is involved in both innate as well as acquired immunity. And in the complement system in acquired immunity works only when antigen-antibody complex is formed. So once this antigen-antibody complex is formed, complement system, these are the proteins, and they are also flowing in the blood all the time, but in an inactive form. They get activated only when this antigen-antibody complex is formed. So once this complex is formed, complement system proteins, they get activated and they secrete perforin. So once the perforin is secreted by the complement proteins, they will form pores on the cell wall of the bacteria. As a result, the bacteria will die. So this is the concept behind humoral immunity. Okay. Now the next we will see cell-mediated immunity. cell-mediated immune system or CMIS, it is associated with T cells only. Okay, now we will see what happens in cell-mediated immunity. Again, when a pathogen, this time we are taking an example of a virus. When a virus enters into the body, it will first encounter with the antigen presenting cells, for example macrophage. That is common in both humoral immunity as well as cell mediated immunity, because any pathogen that enters the body, it will encounter with the antigen presenting cells only. So here we are taking an example of a virus. Say this is a virus. It encounters a macrophage. Now the same thing will happen. It will phagocytize the macrophage. It will phagocytize the virus. Then phagosome will form. Then the lysosome present in the cytosol of the macrophage, it will get attached to the phagosome and form phagolysosome. Now this lysosome will secrete hydrolytic enzymes that will digest the virus, and the antigens will float in the cytoplasm, just like humoral immunity. But there is a slight difference here. Now this macrophage, what it will do? Since it is an APC, antigen presenting cell, so it will have MSC2 molecule. So what it will do? It will present these antigen on its surface. Okay. This is the same as we saw in humoral immunity. Now, suppose this virus is attached to this macrophage, but some of the virus, they might enter into the say epithelial cell. So I am drawing an epithelial cell. For example, this is an epithelial cell. If some of the virus gets a chance to enter inside the epithelial cell, so inside the cell, once it gets inside the cell, what will happen? The lysosomes will again digest this virus, but the antigens will again float in the cytosol of this epithelial cell. But this epithelial cell consists of a special molecule which we call MHC class 1 molecule. MHC1. MHC1 molecule is present in all nucleated cells, almost all nucleated cells, except antigen presenting cells, because antigen presenting cells consist of MSC class 2 molecule, but all other nucleated cells consist of MSC class 1 molecule. Now MSC class 1 molecule, what it will do? It will take these antigens on its surface and represent the antigen. Now come to this point. This macrophage, now it is called antigen presenting cell, since it is presenting antigen with the help of MHC class 2 molecule. Now it will come in contact with the helper T cell, just like humoral immunity. So helper T cell will attach to the APC. This is TH. Again it has TCR and CD4. Okay. Now this TH will secrete IL-2 and interferon gamma or gamma interferon. With the release of these two, interferon gamma and IL-2, cytotoxic T cells, they start proliferating. Cytotoxic T cells or killer T cells. Killer T cells will start proliferating. Killer T cells. Now these killer T cells have CD8 molecule. Killer T cells consist of CD8 molecules. Now this molecule, these killer T cells will go to this epithelial cell, and here it will attach to the to the antigen through CD8. Okay. Now after this attachment, the killer T cell will secrete perforin. So these killer T cells will attach to the MSC class 1 molecule and the antigen, and the killer T cells will secrete perforin. And as we know, the perforins, what they do, perforins are chemicals which form pores on the membrane of the cell. So the infected cell will have pores once perforins are secreted by the killer T cells. So after the formation of the pores on the cell will die. The infected cell will die. So that is the mechanism of cell-mediated immunity. Okay, so we just summarize here. A virus enters into the body. It encounters first with the macrophage, which is antigen presenting cell. It will phagocytize the virus. It will then break the virus into pieces, and the antigen will float in the cell cytosol. Now we have MSC class 2 molecule. MSC class 2 molecule will take these antigens on its surface, and this APC will encounter T helper cell. T helper cell, once it forms a complex with the MSC class 2 molecule and the antigen through TCR and CD4 molecule, the T helper cell will secrete interleukin 2 and gamma interferon. Once TH cell secretes interferon and gamma interferon and IL-2, cytotoxic T cells or killer cells will proliferate, and once they form a number of copies, they will reach the site of infection, and they will attach to the MSC1 molecule and the antigen through CD8 protein. After this complex formation, the cytotoxic T cell will secrete perforin, and the perforin will form pores on the infected cell, and the infected cell will eventually die. So this is the concept of cell-mediated immunity, and in the next section we will discuss about the different types of immunoglobulins, okay, antibodies.